So the next thing I'd like to talk about is variation formats. When you're working with genetic variation, you'll see it in numerous different styles. One of those styles is just prose, which hopefully you can interpret, but some of the other styles you might see are genomic coordinates. So this expresses the location of the variant on the reference genome. And this is quite nice because it's unambiguous. If you state which reference you're using, if you remember last week I talked about GRCH37 and GRCH38. So long as you state which reference you're using, this tells you everything you need to know. RSIDs are identifiers assigned by dbSNP. They are brilliant because they are unambiguous. The location and the alleles are stored in the dbSNP database, and it gives you location on multiple genome assemblies as well. Another one I'm going to talk about in more detail is HGVS. So this expresses the location of the variant in terms of a transcript or a protein. So the example I've given here gives an ensemble transcript ID, a C telling you it's a cDNA type, and then tells you the variant change. The big problem with HGVS is ambiguity, and I will talk a bit more about these if you don't specify isoforms or versions. Another one you'll come across if you're looking at somatic variants is cosmic IDs. So these are COSM and then a number, and these are assigned for cancer variants. Again, they are unambiguous. The location and the alleles are stored in the database. So an RSID refers to a specific variant, and it gives information on the locus and the alleles. Further data, such as allele frequencies, can be attached to RSIDs. So if you've identified new variants, if you've done a study that gets variation data, you can submit it into dbSNP, and every variant that you've submitted will be assigned an SSID, which stands for Submitted SNP. What then dbSNP do is they curate together submissions of the same variant locus to give a reference SNP, an RSID. So if you search for an RSID, you'll find that it's got numerous SSIDs, and it will tell you what study they were found in, and it will give you the alleles that they found and include the sequences that they got. So another format that you'll come across a lot if you're interested in looking at variants by locus is VCF format, and this is the standard format you will get at a variant calling pipeline. If you run something like GATK, you will get VCF. It must give information about location, and it must give information about alleles, but it's got extra columns that are optional that give you a lot more information and can include things like genotypes, frequencies, and quality scores. So this is a really simplified version of a VCF file, and it consists of the first column is chromosome, then position. It is a tab separated file, so you can find that these columns can get out of sync a bit, and it's easier when I sort of put it into this table format. So we have the chromosome, the position on that chromosome, the identifier. If you don't know the identifier, if it's just come off a variant calling pipeline, this will just be filled in with a dot, the reference allele and the alternative allele. If we actually look at a full VCF file, and this is one that's come out of 1,000 genomes, they are big and busy and messy and not the most obvious how to follow, but I'll give you some tips on how to work your way through. You will get different things in different VCF files. So I've just condensed down the info column here. There's loads and loads of information there. But we have things like the quality score and the filter. Again, if you don't have these will just be a dot. And then we have a format column which says GT, which stands for genotypes. And this is followed by identifiers for individuals. This comes from 1,000 genomes, so these are the 1,000 genomes identifiers, and it tells us what genotype these individuals have at this particular locus. So the genotypes are given in the format 0 means reference, 1 means the first alternative, and then 2 or more means the second or more possible alternatives. So if we look at this example line given here, we have genotypes of 1, 1, 1, 0, and 0, 0. And we know that our reference is C and our alternative is T. So we can say that this first individual has the 1, 1, the alternative, so that's T, T. The second individual has alternative reference, so that's T, C. And the last one has reference, C, C. The genotypes I've given you here are phased. That means they have a pipe in between the numbers. The 0 and the 1 have a pipe in between them. And that means we know which of the 2 chromosomes they come from. We can use this to calculate LD, and we can do things like determine the actual genomic sequence in this person. So for example, the haplotypes, the transcript haplotypes page I showed you before is calculated using these genotypes. If you ever see a VCF where it's got a slash rather than a pipe, that means we don't know. And this can be useful. So if we take 2 variants that are next to each other in the file, and this individual HG1879, they've got a 1, 1 and a 1, 0. We know that in one of their chromosomes it's reference and then alternative, and on the other chromosome it's alternative. So this would be, so then our reference here is 1, so we've got our 1 reference refers to this, alternative refers to this T, our 0 refers to T again, so 1 copy has got T and then T, and then we've got 1 and then 1, so that's alternative, T and then C, so we've got another copy which is T, C. So I left the info column for later because there is so much data in the info column. There are loads and loads of fields that you can put in the info column, and these are actually defined by the header, and you can put pretty much anything in here. There's some standard fields, and it's worth referring to the documentation for these so that other tools and things can read it, but you can put in other things that you want to see. So if we look at for example, AA equals G is one of the things that's defined in this info column. So if we look through the header at the top of the page, we will find ID equals AA, and we find that its description is ancestral allele, and the format is the ancestral allele, reference alternative, indel type. So these are if there are actually variants in other species. And so we can see that in this case, the ancestral allele is G. So you get quite a lot of messy data, but messy kind of descriptive, but once you've worked out what it means, it's very easy. A second example, we've got AFR AF equals 0.3691, and up in the info data we find that this stands for allele frequency in the African population. So we can say that the frequency in the African population is 36.91. There are full specs. If you are working with VCF, it's worth checking out the full specs, which are published by SAMTools, and it gives information on how you can encode things like short, indels, simple and complex, structural variant, and some of the standard things that can be included in the info column. So now I'm going to talk about HGVS notation, which describes variants in terms of the CDS and the proteins they fall in. So we've got a CDS example. So it consists of the transcript identifier, a colon, C for CDS, dot, and then the position of the change, and then the change, and a protein example. So it's the identifier of the feature they fall in, followed by the change, and they do have, again, detailed documentation available. So this is just summing up the CDS notation, ID, telling you it's CDS, telling you the position, and telling you the change. The protein identifier notation puts it in a different order. So we've got ID, P for protein, and then we have reference amino acid, position, and alternative amino acid. So it does look a bit different. There are problems with using HGVS, and a lot of this is actually to do with the way people use them. If you use transcript and protein identifiers with version numbers, this is good because it is unambiguous. If I put in this identifier here, it tells me which transcript it is, and it says it's version 7. So I know exactly what it is I'm looking at. I can find this again, and I know that I'm looking at exactly the same thing that the person who originally wrote this HGVS was looking at. The problem is there's lots and lots of people who don't do this correctly. So taking this example, this people will often use a gene name rather than a unique identifier. So they haven't said which database they found this gene sequence in. Different databases will have different sequences for it. They haven't said which transcript it is. And they also haven't given a version number. So actually this arginine 145 could be, if I looked at a different version, could be arginine 146. But I don't know from what it is they've told me. And if there's 2 arginines in a row, that doesn't really help me. Another problem you'll see is repeat shifting. And there are different sort of methods for writing how you put repeat. So if we look at this example sequence given here, where we've got 4 Gs in a row and the alternative is 3 Gs in a row. The question then is, which G is deleted? Is it this one at the beginning? Is it this one at the end? Or is it one of these ones in the middle? It doesn't actually matter because it's the same thing, but there are different ways to write it. So in VCF, you would write this, for deletions, you actually write the base before. So you would say, right, position 7, which is the base before, is a C. So you'd say position 7 is CG, and it becomes just C. So we've deleted that G. HGVS shifts to the opposite end. So this is actually genomic HGVS. So you would say the chromosome number and then G, and oops. And we would say that this G, position 11, was the one that was deleted. The problem with doing this is if a variant is referred to by 2 different locations, then it might not be identified as the same variant. So somebody might do a study looking at phenotype, search for, and sort of say this phenotype was found at this locus. Somebody else would do a study and identify that the frequency of this variant in a different population, and these are actually referring to the same variant, but they might not be linked together in the databases. Ensemble deals with this using normalization, but if you're doing this in a manual fashion, you might not think to look for this kind of thing. Another thing is, once you get out of simple things like amino acid changes and things found in coding sequences, HGVS gets very, very complicated. Anything in an intron, things like insertions, these can be very, very complicated. There is documentation on it, and it's quite hard to follow. So I'm going to move on and talk about the Ensemble VEP. And the Ensemble VEP knows how to deal with all these different kinds of formats. And what the Ensemble VEP does is it takes your own variation data, which could be coordinates, it could be VCF, it could be HGVS notation or a list of variant identifiers, and it tells you which gene they hit and what consequences they have on those genes. So you can start to understand what might be phenotype causing and things like that. So the VEP does this, and there is an online tool and also a standalone Perl script. So I'm going to show you the online tool, and we're going to have a practice at using that in today's presentations, but I'm also going to give you a quick demo of the standalone Perl script. It requires quite a lot of downloading stuff and installing stuff. Plugins add extra functionality to the VEP, so they can extend, filter, or manipulate the output. They often make use of external data, and some of them are available on the web tool. All of them you can use with the script. And there is a paper with a lot more information about the variant effect predictor. So we're going to have a look at a VCF of variants from whole genome sequences. We're going to run these variants through the VEP to identify which genes were affected.